Hi everyone, thank you for coming. Another Friday to the seminar. Well, today I think that I don't need to explain a lot who is he's going to present. Well, we have Eneko, that he's working in Mars Group, like a postdoctoral researcher. And I only want to say that if we don't get any volunteer for the December 15th, theoretically today this seminar will be last of this year, so enjoy it and go for it.
No, it's a lot of pressure. Okay, so, well, I guess maybe Kiska doesn't need to explain who I am, but maybe I need to explain what I do because I have been here a while, but it's the first time I give a seminar here. So I guess many of you don't know what are we working on, and it's what I will explain today. And also, as it is the last seminar of the year, it will be special because there will be a contest. So, with a prize at the end. So there are several pictures. You will see pictures of fish because I like taking pictures of fish. There are a lot of pictures of fish along the talk. If anyone tells me how many fish appear in the presentation at the end, you get a prize. No, okay. This slide does not count. Videos do not count because there are some videos, and also silhouettes or drawings of fish either. So they have to be real fish. Okay, see that. Okay, so yeah, the talk is about fish movement and connectivity because we are working in our research team. We are especially focused on marine fish, on coastal fish, and also with very related to the fisheries, to either traditional fisheries and also recreational fisheries. Because, sorry, as I guess we know, we live on some islands, so we are completely surrounded by water. And that means that the population in the Balearic Islands is quite special because they live very related to the sea, because you always have any coast. So, and indeed, we have a very diverse biodiversity in our coastal areas. Okay, this is not a fish. And so we have a lot of a diversity of fish, of a lot of species, but they don't only contribute to the biodiversity of the islands because they are also important resources. There's another fish here for either the artisanal and the recreational fisheries. Here you see a recreational fisherman. This is a scientific fishing, not one exception. So they're very important economical values of those fish too. So we need to manage them because, as we know, this fishing has an impact. And indeed, here in the Balearic Islands, we have a very nice panorama of management because here the fisheries directorate is very active in managing these resources. And as you see, we have a lot of marine protected areas around. So we have right now 12 marine protected areas in the islands and a national park here. I guess you know many of them. And not only that, is that also if you see the dates, this is when they were founded. So the oldest one is from the 80s. So in the 80s, they're having this active management of fisheries. But if you see some of them, they are really, really recent, like last year. And also some of them have had modifications in the recent years. So it's something that here in the government are really take care of it. And I think we are the region in Spain with more protected areas. And also for us as researchers, this is a very nice setup because we have a lot of protected areas quite close from each other, so a lot of research can be done. And it's where we are focusing a lot of our research. So as I said, to manage them, there's a lot of management efforts, but also we need a lot of information to manage the populations of fish we want to manage properly. And part of the information is what we work on, and it's the movement of fish. Because we need to know where fish are, which habitats they use, how they move from one side to other, in order to make this management effective. And this is what we do. How do we do it? So we use this technique that is called acoustic telemetry. This is another fish. And this is the last time I see now. From here, you can start counting them. And this is not a fish because it's a sea. So we use this technique that is called acoustic telemetry, where what we do is we implant some transmitters, some electronic devices on fish, and then we place some hydrophones or receivers in the area, So when the fish gets close to a receiver, we know that the fish was around here. This is the basic of the techniques, and we will see that it has some development later. And it's a technique that is quite popular, and in the last years has become an important research topic to monitor its movements in the marine environment. Here you see this is a bit updated because it's a science paper from 2015, so it's only arrives to 2014, and it's a number of papers that are using this technique, and especially used in fish, because in other fish that they get to the surface like mammals, so they use the satellite, which is another technology. And this is a bit more updated article, and here you see that these numbers are still increasing. So it's as it goes together with the technical innovation of these techniques that people are applying them more and more to do research. And why they are super interesting? Because they are super diverse and you can adapt it to study whatever you want. So you have a lot of different sizes of transmitters that you can implant, since big ones that the batteries, they last a lot, even to tiny ones. So you can also now tag juveniles or small fish. And also in those transmitters, you can have a lot of sensors. So not only you don't only know where the fish is, but you can get information about the environment of the fish. So here you have just some examples of the most used sensors, like temperature sensor, you can know the temperature at which the fish is swimming, pressure sensors to know the depth, acceleration tags to know the activity. And with all those sensors, what you can do is to study a lot of different aspects of the biology of the fish. So it's an example from my PhD several years ago. Here, this is an experiment we did in Catalonia. We tagged some common tentex. It's a predator here, like very common in Mediterranean Sea. It's a top predator. So here we tagged several individuals, and here where you can see is where the fish were in depth with the bathymetric. So as you see, during the most of the year, most of the fish, they stay between 20, 40 meters. Something happens here in May. But then what we saw is that suddenly in summer, they started doing this kind of migration. So all the fish that used to stay like deep, they started going up and down. And we checked it with the temperature. We saw that there was a high correlation with the thermocline. So this means it's a temperature around the year. This is in summer when the water is stratified. Here you have the thermocline. So it seems that these fish, when the thermocline rises and the water is cold, they just go upper. And when the thermocline goes down, they go back to the preferred depth, to the habitats they like. So we can get a lot of information on the thermal ecology of the species with this kind of data. This is an old example to illustrate this. So all these studies that you can do on the biology, they can be very basic on the biology of the fish, on the ecology, but then they can be also very applied because they can help us to answer a lot of the main issues with the management nowadays, from fisheries management, MPA design, that I said before, also how the biodiversity responds to human impacts, habitat restoration. So it's very versatile and it can be used to answer a lot of applied questions. Okay, so this is what I explained, like the basics of acoustic telemetry, but they're like, we can apply them in different ways, and there are like two main ways. There is something called high resolution acoustic telemetry, where you can get a lot of more detailed data on the movement of the fish. I will explain it later. Or also you can use it to, like they are opposite. So if you do one, it's difficult to do the other one. So there are like two applications, and I will explain now. So let's start with the high resolution acoustic telemetry. And I will explain it with a practical study we did. We started this study in 2019 in collaboration with the Leibniz Institute of Freshwater Ecology and Inland Fisheries in Berlin, who lent us all the material, you see, because one problem of acoustic telemetry is that it's quite expensive. So what we did, we put a lot of receivers in a small area here in Palma. This is the Bay of Palma. Is Cap and Rocat will be here. The delta. I guess most of you know the delta is here. So this is a sandy patch. This is like 600 meters. So here what we did, we put a lot of receivers to do this high resolution acoustic telemetry. Because with this system, what you do is that when the fish they move around, you are able to triangulate where the signal comes from. Because usually with acoustic telemetry, you only get presence absence information, so you know if the fish is there or not. But with these techniques, if you put enough receivers together, you can triangulate the position of the fish. So if the fish is here, the signal will arrive to several of those receivers at the same time. And with these differences, as the GPS system does, you can get a super precise estimate of the position of the fish with less than 1 meter of error. So really precise. So and we studied the razorfish, the raor. As most of you know it. It's a very emblematic species here in the Balearic Islands. People, especially recreational fishermen, they love it. And there is a fishery closure during the year, and it opens the 1st of September. And it's a kind of national holiday that day because a lot of people they just will get a free day to go and to start fishing this fish. And with some studies that Pep did, in just a few weeks they can take out 70% of the population in just 2 weeks. So it's really intense. It's a very key species in the Balearic Islands. And we tag them with those really tiny transmitters. You see, this is like some less than 5 millimeters. So it allows us even to tag both males and females. You see, there is a very clear sexual dimorphism with the females having this white spot here. So we were able to tag 300 individuals at the same time and monitor them simultaneously. And here I have a video of how this technique works compared to catch a fish. These fish don't count. Okay. So then we put them on a tank. And then I didn't explain that, but the transmitters, they are placed in the peritoneal cavity of the fish. So we anesthetize them, and then we do a small surgery, making a small incision here. We put the tag, and then we stitch it with some surgical stitch. Here we have, this is not because of the surgery, it's because it was COVID time. And now it's similar from all times. And then we do some surgical stitches, which is sometimes a bit tricky. You see here, you see that the boat is moving quite a lot. Sometimes the pulse is not really good. It works, and then the fish they recover quite really quickly, and so then at the end we release them. So what kind of data we get with this? So this is, you have here an example of 1 day of trajectory. So we can get a lot of trajectories, really detailed trajectories of many individuals at the same time. Here you see in pink the females, in green the males. So we can get a really nice view, kind of reality mining approach of what is going on in this population. And here you see they disappear. And I guess you already know that is because this fish sleeps at night, they burn in the sun, so we don't get any detection. So they disappear during the night, and then during the day they appear. So then with these trajectories, a lot of work can be done. For instance, here we apply some hidden barcode models, some statistical models that allow us to split the trajectory in behaviors. So we can distinguish between the home range behavior when the fish, the areas staying in some areas, or when they are doing some excursions, they are being more exploratory. We also describe the social structure of the population. So here you see that the territories of different fish. In green the males, in pink the females, and here something can be seen from this picture. And if you see the males, they don't. It's each one. It's a very territorial species, and each male has its own territory, and they don't bother the other males. So each one is in territory. And the females, it seems that they have a similar structure, but they overlap with males. So we applied some, and here also we could also measure the variability, the behavioral variability in the home range sites. So here we see that different individuals, they don't have the same home range. So they also differ. There is a big relationship with the size of the fish. Here the fish are ordered by size. The smallest one was like 10 centimeters a female. The bigger one, 23 centimeters, I think a male. So here there is a relationship between the size of the fish and the home range. So the bigger the fish, the bigger the home range. Not in males. Then when we check it in males, it seems that when they become males, the home range becomes a bit more stable. But there is this relationship. And also there's a lot of variability within each individual. Each bar is one individual. So there are individuals that they have bigger home ranges than individuals from the same size. So there is a variability here. And also we apply a social network analysis to see these interactions, because as we have these super detailed trajectories, we forget when the individuals were close to each other, were interacting. Well, we say interaction, but it's more an association, because we don't know really they were really interacting, but they were really close to each other, so less than 1 meter apart from each other. So with this, we constructed this social network, and we could see this kind of structure of the population with a kind of arm like, no? So there is a male that is created with some females, but males, they don't interact with each other, and females, they interact inside the areas, but they don't interact with different areas, and then something happens also. Here you see a big bunch of females. I will explain it later, but generally they don't interact with each other. So there's a kind of avoidance of individuals of the same sex and interaction between individuals of different sex. So then this was at the beginning of this experiment. This has been a beginning of a long research line with a lot of works that have been derived from these trajectories. In our team, Pep has led 2 projects that have been focused in this kind of data, from the national plan from the Ministry of Science, the CLOCKS project that ended last year, and the raor project that is ongoing right now. In this one, what we analyzed was as the fish gets in the sun, it has a very clear day and night activity pattern. So it's what especially Martina and her PhD analyzed. And now in the raor project, we are trying to apply all this knowledge to which consequences they have for fisheries management, all these differences we see in behavior. And also here you can see a lot of work have been derived from this work in different topics like the chronotypes, these day and night cycles. I don't explain them because most of them, I think they have given, a lot of them, they have given a seminar right here. So, but it's just to show you that different things are going on with this. Like for instance, also Aina is working with the good microbiome and seeing the interaction between the good microbiome and this the behavioral diversity we see in the nature. And also Jordi, who's not today here, but he worked on seeing how the environmental drivers, they can affect these different differences in behavior. Also Marco is working with those trajectories, finding new ways, applying artificial intelligence to segment the trajectory, so a lot of work. And do you remember the females that I said that they don't interact, but sometimes they interact? Also, this is something, it's a curiosity we observe, and it's that in certain days of the year, they start doing this kind of collective movements. It seems like a demonstration, but only females, and not males that were not included here. They were not participating in these kind of excursions, and in those days, almost 20% of all the females with that were doing this kind of behaviors together. And we don't know what's going on here. We suppose that this may be a commensalism behavior. So it's what I believe, that because the kind of movement here in the close to this is a Posidonia meadow here, close to the Posidonia and up and down, it resembles a lot to the trajectory we have on some rays. So we think that there is some rays eating in the bottom, and the females just go behind eating when they move. And it's just before the spawning, so maybe they say, okay, we need to eat. Let's follow the race, but we don't know, it's something we need to explore because it's quite curious. And it only happened during 1 week. Then after that, they were not moving from the territory. Okay, so those were the examples of this high resolution telemetry and how we apply them. And now I will talk about this large scale telemetry, where instead of having a lot of receivers together, what you do is you put all the receivers more spread out so you can cover larger areas. You don't get these precise movements, but you can cover really, really big areas. And for that, what we have developed is the Balearic Tracking Network. That is an initiative that started here in Imedea of having receivers in water, basically trying to cover as much of the coast we can. It's an open initiative, so it's also open to other entities that if they want to put a receiver in the water, they are also welcome. So then together we can share the data, because at the end, here is, for example, those are in green is the receivers we have in the water right now. Part of the Balearic Tracking Network. Here we have in yellow some new arrays we are using for some razorfish studies this year. This is from the project EPAMAR, the receivers we put this year. And also here are some receivers that were put by the people in COFIP, that they are working with rays. So they can collaborate, they put receivers, they are included in this database, so then we can share. And they are tagging fish. So the nice thing is that as we share the data, if any of the fish they tag is detected by any of our receivers, we can share the data and give the data to them and vice versa. So if any of our fish go here, we share it. So it's a nice collaborative environment to work. We work with something that is called the Open Protocol, where we also participated in its development and establishment, which is because before they were each receiver and transmitter, they were like the wires of the cell phone. So each, like the iPhone wire does not work with the Android one, and that was a mess. So if you were tagging fish with one brand, other brands will not detect them. So with this development that was developed from the European Tracking Network, that is this group of people in Europe working in this, in telemetry, like, okay, we need to be compatible always. So if we make sure that our fish will be tagged, whatever the brand we are using. So all our array is based on the Open Protocol. And so our receivers are like kind of lighthouses in the sea, so that are just observing what the fish are doing. So we put some light under the. And also is in the European Tracking Network, they have also a database. So all our data is also uploaded to the database of the European Tracking Network. So also if people in Italy, in Catalonia, in other places outside the Balearic Tracking Network detect our fish or vice versa, we will also know. And it's part of this, now it's also a lot, there are a lot of integrated tracking networks around the world where people are doing these same things of putting receivers and sharing the data. And indeed, like as a part of the European Tracking Network, we just hosted the European Tracking Network Symposium 1 month ago here in Palma with all the researchers that are working in Europe using these same techniques to answer similar questions. And in this context is where also we got this project funded by this biodiversity call from the government of the Balearic Islands, which has the objective of studying these movements between different protected areas, to study the connectivity of the protected areas based on the movement. So here we will just consider the connectivity of individuals moving from one side to another, because it's also a very important part of the connectivity, because it's also when people can fish them, because if fish are inside the protected areas, usually you can fish them, but when they move, and in this traveling from one protected area to another is when people can really extract those resources. And so we have there are several more objectives of starting this long-term monitoring of species to characterize the space use, the activity and variability of behaviors, quantify how effective is this array that I showed in the beginning of protected areas in really protecting those species, because now we can measure how much time they spend inside each one, and to study the measure the connectivity, and also try to develop new management recommendations focused to those species. And also promoting a bit the Balearic Islands as a key point to study the movement of fish, as we are in the center of the Western Mediterranean Sea as a kind of hub for studying these species. So here now I will show different species we have worked with in those years. The first studies we started here, like with the Balearic Tracking Network, were focused on the grouper. You know, the grouper, I guess, is another emblematic species. They are really long-lived. They can live up to 60 years. They are slow growth, very territorial, but also a good indicator of the quality of the environment, because it's a good indicator of low fishing pressures. So we started these studies in El Toro, where there's the largest population of groupers in this area, and our objective was to start a long-term monitoring of those fish. The next thing of the tags also is that as they are big fish, you can put big tags on them, and also with big battery life. So we can follow them up to 11 years, what they do, because it's known that they are territorial, but usually the studies, they are focused in some months, 1 year, 2 years at most. Here we can follow them for a more significant period of time. So we started with this small array of receivers. It was the beginning to then the other array I showed before. I have a video of these studies of how we work. So here they're like big fish. Also, in order to catch them and being selective, our catching technique is diving, scuba diving. So it's me. Here I have a hook with a calamari. We put a calamari here, and then we wait to see if they eat or not. And then when they eat, here is the calamari, and here behind. So now here, Marco comes with a bag, and we kidnap the fish. So we put it inside. And this bag is a very nice improvement to this, because once they are inside the bag, they get really calm because it's dark, they don't move, and then you can just take them to the surface little by little. Like here, it's inside here. And then once on the boat, we put them on anesthetic. So here there is an anesthetic dissolved in the water. So when we put them, the fish gets slept, and then we can work with it easily. We use this big, it seems like no, but it's better to use big hooks so they don't eat them. So it stays in the mouth. So that's why we use this huge one. And then we put them in these surgical tables. We have improved it. Now we have an immaculate fancy one. And then we do this operation. So we cut it, then we put the transmitter, we stitch it. And then also we put a external mark in order not to catch the same fish twice. And also if any fishermen they catch it, so we know it. Then we put it in clean water so they recover. They recover really like in 2 or 3 minutes they are awake again. And then at the end we release them. Yep. And they go happy. So this grouper has this tag that for 10 years will be emitting this sound and these signals that we will be able to detect them. So this is the data we got in here with those. We have a small number because it's not really, they are very smart, those big groupers, so it's difficult to catch them. So here you have is the detection pattern since we tagged them in 2021 until the last receiver download in February this year. And these bars are like detections per day. So you see, we detect them constantly through the year. Here, it's another problem of the things that the hydrophones, they use batteries, and the batteries stops at several months. So here, we were not able to go to change the batteries, and we lost some data in between here. So it's not that the fish left, it's that the batteries, the receivers were there. So here, something surprising that we were not expecting is that as you see most of the fish, they are not longer in the area. They have departed. Something we are quite surprised because they are supposed to be really territorial. We're expecting to have them there for a lot of years, but for now we have only this one, and those that were tagged later in Margrats that stay are here. The others, they have departed from the area. And it's also another problem of this technique is that when the fish leave from the array, from the receiver array, you don't know where the fish are. So maybe they migrated to some rocks outside here. Maybe they were fished, or maybe they were poached, or maybe they left and maybe they will come back. I mean, the battery should be running for 10 years. So maybe this year when we change the batteries, we see that some of them come back. But, well, there are a lot of questions here. Once you get, because usually all the studies with groupers, imagine that we're not always in this period of time, like only 1 year. And in 1 year, they are all territorial, but then the longer you see, things happen, and the less we know. And also inside this project, we also started a project with juveniles of groupers in Menorca, in La Isla Aire. It's also a protected area, quite recent. And here, what happens is that there is a huge density of juveniles. It's the most abundant species, is this one. It's a very rare species overall, but here it's a very nice recruitment area. So there are a lot of tiny groupers, but then they are not big ones. They are not there. So the biggest one you see is like this. So you have thousands of juveniles of 1 year old or 2 years old, but then you don't see any 3-4-year-old fish. So we wanted to check if they were predated or they were living or what's going on with those fish. So we use those transmitters that are super tiny, are the tiniest transmitters that are made with this technology, that they have a mortality sensor. It was something very preliminary to see. There are some studies that they are applying it in salmons and it seems it works. So it has a tilt sensor, like our cell phones. And when the fish is alive, it's supposed to get a constant tilt. But then when it's predated and it goes through the intestinal tract of a predator, it will start running. So this triggers something inside the tag and it changes the code. So you know that the fish is dead. So it's okay. Right, they are tiny. It can be interesting to see if those fish are predated or not. So this is what we did with our colleagues from the Spanish Institute of Oceanography and also the colleagues at the La Mole station. We catch those tiny, super nice individuals. We tag them, as I saw before. We release them. This one is not happy. You see, this is a bit angry with us. But it seemed that the operation went well. So this is the individuals we tagged. Indeed, the individuals we tagged with the mortality sensor are those that we tagged them this June. And this October, we download the data. And we have all other individuals that were tagged with conventional tags, not mortality sensors. And also, surprisingly, most of them, they don't left any signal. So we don't know any, didn't detect them. So maybe they died after the tagging or they were predated. And also the thing is that all the signals were predated signals. So all of them were supposed to have been eaten. But then when you check the data, well, this one also another one was tagged with a pressure sensor. So this one we saw that left. So this was one receiver, and then here, as you see, it went to another receiver, another color, and deeper. So it seems that swam deeper. So they left the area. And the ones with the mortality sensor, I think that something happened with the tags and it didn't work well because we have a fish that has been there for 3 months, giving signals constantly. Here you see this is when it was alive and this is when it's dead, but you see that the pattern is quite similar. So I think that this tag they triggered before time and we didn't get the data we were expecting. Well, but the grouper that didn't move. So I was talking about connectivity. It's not a very interesting species for connectivity because they are supposed not to move. Then we will see what happens with those that disappear from the tunnel. But then we are also working with other fish that are more known to move, that will give us more information of this connectivity, like the common dental. It's another top predator. I introduced it before. Also the barracuda, the sped. I put a video here, so you need to count them because remember videos, they don't count. That they also another predator, they dig these big shoals, but very little is known about the movement patterns. And also the amberjack, the ceriola, is another fish, like a very paradigmatic fish here, like trophy fish. So fishermen they like to get because they get really, really big, 1 meter plus. It's like the species we are working with. And also it's a very important species here with the verderol. That it opens the fishing season in September, that is catching the small ceriolas. So also it's a very interesting species for recreational fisheries. And also we are tagging some other fish that are of interest for more from the conservation point of view, like the batoids, the elasmobranch. So we started this year with tagging a lot of these pastinacas. I guess you know the fish with Xyrichtys pastinaca, which live in the Bay of Palma. There's a nursery area, so they come here to reproduce. And also some other opportunistic species that we were able to catch, like the eagle ray or the battlefield ray. And we did this in collaboration with a local fisherman here that he put a net on the sea and he captured all the individuals for us. But it's in 2 days we were able to tag all those and many other species, not with acoustic tags, but it was quite effective way of working. Yeah, so here he was giving us like not only the Pastinakas but also other species of rice that he was giving. I mean, we were able to catch a lot of them quite quickly, and indeed now Pep is leading a new project, a kind of continuation of this project that I was explaining, also funded by this biodiversity call that will be focused on those species, because we saw that it's a species that is very interesting and very easy to work with, because they are easy to catch. We know where they are. We know where they are in that moment of the year, and we don't know what happens with them. So this is the kind of data then we get with this technique. Okay, talking about connectivity, we have this gradient of different species that can contribute to the connectivity, like the groupers. You see, they usually don't move. Here you see those are movements of individuals from one station to another. Groupers usually they stay where you tag them. With the barracudas, we still don't know, because this is super short time of data, because we tagged them in November, December, and the data was downloaded in February. So we only have 3 months of data. This data will be increased this year. You don't know, but well, we see that there are those movements between one protected area to another. But then the interesting species are these, like for instance, the dentex, that they are able to move quite often from quite far away. Like this is El Toro, protected area, this is Dragonera, Portland Drudge. So they move, and also the amberjacks, we see these big movements from the Bay of Palma to protected area, Bay of Palma to El Toro and to Dragonera. So, and this is just some months of data, so we still don't have the entire data set, but we are expecting to get really interesting movements here. The same with the Pastinakas. This is the nursery area where they come to reproduce, to spawn in May, in spring. So we catch them here, but then some of them they go to the west, some of them they go to the east. The same with the butterfly ray. Well, and this we are now indeed one of the Pastinakas we detected it in some receivers we put in Cabrera. We were able to detect it. And also with this data, we can not only see this connectivity, but we can also study this activity pattern. This is a new method we are developing, where we can, from the detection data, we can know the distance at which fish are from the receiver. So we can know if the fish they move or they don't move. The clearest example is this one, is the Yampuga. This data from a project that Ignacy led on Yampuga, Polyphile Purus, and here we tag this fish surrounding a fishing aggregation device, so a floating object where they aggregate. So here you see that during the night, those bars, the fish usually they stay quite close to the receiver, that means to the floater, and then during the day they have these more extensive movements. The same with the butterfly ray. You see here that it's standing in the sun during the day, then during the night they become active. The Pastinaca is the opposite, stays quite not moving during the night, and then during the day they move. So we can get a lot of information on the activity patterns of this fish from this data. Yeah, well, this is a general map of the kind of product we want to generate, on how the fish they move from one side to another, hoping that will help management at some point. And I think, yeah, okay, so yeah, thank you for listening. Thank you also to all the people involved in all this research, the research team, and also all the people that somehow inspire our work, even if they are not here anymore. Thank you. Questions?
Thank you for this talk. I once I work in an institute where they used what they called ears, is ecologic acoustic receivers thing. Which basically they were using receivers, but not to monitor. They were not tagging fishes. They were using it to capture sound,
The sound,
ecologically relevant sounds, and they use it to estimate densities of different.
Yeah.
Can your receivers we used for
Our
this?
receivers, they only listen in a single frequency, that is the frequency at which the transmitters are emitting. So it does not record all the spectrum of noise. But indeed, with those receivers, we could detect our transmitters because you can get the pins. But no, it's only this data, it only gives you if the fish has been detected there or not. You don't get a spectrogram of sound that you can analyze.
I have a question. Oh, sorry, please.
I have a question about the movement of the females, so they're together. I'm not sure like if I will do the question. So some of the females, they overlap, but not much.
Yeah.
And some of them, they were in the same territory as the males that are bigger.
Yeah.
Are they, well, the first would be, are the females that are moving together in the same territory?
No, as in the video, you can see that they go together from when the group of females arrives to another female, the female joins. So, but they are females from all around there.
And do you know the size? Could it be like a protection mechanism if they are smaller, or if the other female,
Yeah.
the some of them are bigger territories? I assume that there's not much overlap that the females have themselves.
Yeah, the females, yeah, they are very territorial. With females, it's something we want to check if there is in this territoriality also, if it is something related to size. Because we see that usually the females they don't overlap. But if that small females, they overlap more with large females, and this confrontation is maybe more about between females that are in the same size. Yeah, it's a kind of hierarchy. It could be like a kind of hierarchy where you have the males that are getting the heteropatriarchal society. Where the males they have their territory, and the females they also fight to each other. And the smaller you are, then well, you are small. I don't care about you. You don't need that much. You are not going to compete for the male for reproduction, maybe. But in these excursions, we don't see any pattern of size or only sex, only females. But there are females from different sizes. Yeah,
I have two questions. The first one is about the diameter when you can get the signal, the receptors. How big can be the area?
For the ones we use in the Balearic Tracking Network, we have estimated it in up to 400 meters. You have a good detection, a good probability of detection, because at the end it's a kind of a model of probability model of how many of the signals you will detect. So you don't detect 100%. The farther is the fish, the less you get. And so up to 400 meters, we have estimated it that at least we get half of the signals. That's supposed to be a good detection range. But for instance, it also depends on the power of the transmitter. For instance, the transmitters we use for the razorfish, that they are so tiny, this range is lower. So we have a detection range of just 50 meters. That's why we needed to put so many receivers together.
And the second question is about the groupers. So in Menorca, you have a lot of small groupers.
Yeah.
And I've seen that in Catalonia, in the south of France, they have a lot of groupers, but not any
Yeah.
juvenile. Is it maybe linked that with the currents, the juvenile and the larves?
It's a very interesting question. Yeah, and it's a mystery. No one knows, because in Catalonia it's true that I was involved in the monitoring of the parks in Catalonia, and people that have been there doing that since the 80s, they have never seen a grouper juvenile. But you have huge groupers, huge biomass, higher than in El Toro, indeed. But no one has seen a small grouper there until this year. This year they have seen for the first time tiny groupers like juveniles and recruits. So maybe it's also related to temperature. So temperature is colder up there, or maybe recruitment is more related to warm temperature. But no one knows where those groupers come from.
Because I've seen the street that they mark also groupers there, and
Yeah.
they're not moving around the
Yeah,
river,
no.
you know, they're reproduced there.
Yeah.
So I guess the larves are getting with the current, and then the recruitment is made when they
Yeah.
make video.
Yeah, maybe could be. Marco asked it, yeah.
So I wanted to go back to the territorial bits. How are the territories established and actually maintained? Because on land, right, you can, I don't know, pee in a corner or make put some marks down, but in water, these are big territories for a small fish, and yet you don't see much overlap at all.
Yeah, it's a good question. We don't know really if there is any chemical marking, or I believe that it's physical. It's a physical. So they will be always patrolling the territory. So when one enters, you will have a kind of but it's a super short interaction that we don't see it because it's just fighting, and so each one has its own territory. Because we have seen in some cases that we have recaptured some of the fish, and we take out one fish, things happen there. So they redistribute the territories. So if you take a fish, the males that are surrounding will just extend their territories, take that area too. And sometimes there are some males that they don't have a territory, they just wander around. And we have one case that we fished one of the males. And then that same day, another male had presented the same territory as the male we captured. So they just, okay, this is an empty spot. So I think there is, but it's interesting. And we also wanted to try to get a kind of model to see how this, because they are very stable, but they are kind of fluid. So very slightly. So I think that they are this, so the borders are not really hard. So maybe that depends on the neighbor, if the neighbor is there, if the neighbor is active or not. But it's a super interesting topic. Yeah, I think that was a question. Eugene? Okay.
You had, coming back to those interactions between males and females, you had one individual there which seemed to have a lot more female interacting with a lot more females. What is the story there?
Yeah, no, indeed.
Like that, there was one kind of.
Yeah, no, the thing is that
individual which seemed to have.
the thing is that here we didn't tag all the individuals. So there are individuals that are doing things and we don't see them. So it's what we have to indeed there are some areas that you can see that I'm quite sure that here there is another male that we didn't tag. Maybe here there was another male. So I guess that here we just have a partial view of the population, and the same with females. We didn't tag all the females, but here, I mean, indeed, maybe those 3 females, they are part of another territory. Like here, you see those ones. Here we fished because how we fish, maybe we fish more intensely in this area, and we were able to capture better the harm than in here. But I believe that this is full of females that we are not seeing. But now, okay.
Now I have a question.
Yeah.
Have you considered, since you are, okay, just apart from the fish mark, like making a bootstrap statistics? I mean, like resampling the one that you have and try to understand how different is the statistics of the subgroup respect to the big group that you have, and try to understand, what's the error associated with the subsampling?
We didn't did it really. I mean, for the network analysis, what we did to see if those interactions were significant or not, what we did some kind of randomizing of the. So what we did was, okay, as we know the area where the territories are, we just redistributed the territories randomly and see if the interactions we get, if individuals were randomly with the same trajectories, but just to moving this territory randomly from here to here, this one here to here, to see if it's a random pattern, if those interactions are the same. And what we got is that the interactions are lower between same sex than if it was a random distribution of individuals. I don't know if it's more or less what.
Well, that's not, it's more related with the fact that, say that the problem is that you didn't mark all the fish. So maybe you can estimate,
Yeah, it's what they're taking as a group and
what's what you did.
Do you have, especially in the large scale movements, do you have temperature sensors there in those places? And do you plan to try to relate the movement of fishes with temperature?
yeah, indeed.
Especially with marine heat waves?
Indeed, yeah. I mean, for this experiment, or you mean
The large scale?
The large scale, yeah. Indeed, each receiver has a temperature sensor. It's measuring constantly, and it's also one of the things you want to explore. What happens when the temperature is really high? Indeed, what the first example I showed, it was quite interesting because in this kind, it seemed that the Yeah, indeed, it's really interesting to see because here it seems like they didn't like these cold waters, even if it is their preferred habitat is here, but they go up. But here it was not a really hot, it was a normal summer, like 24 degrees.
Which year?
It was
2021?
No, it was 2009. Or 10. So it was interesting to see for this kind of analysis can be done with all the species to see what happens when you have a big mass of really warm water here. If they move to another area, they go deeper. What happens? It's one of the things you want to study.
So in summer they want warm water, but in winter they are okay in all the.
Those I mean because they can choose in winter
No,
is the
but
temperature
they
they
are
have.
deeper.
In summer it seems that, but it's what I'm wondering because now we have reached here 28 degrees. So what happens when this is 28 degrees? Maybe they don't like that much. They go. Here we were relating it also as they are active and they are predators that they need temperature. I mean, the metabolism is related to the temperature, so maybe they needed this warm water. It's better for them to catch. It also has been described for many other, like the tunas and many species that they do this kind of.
What's the temperature in which they are most of the time? 18?
Yeah, it's not the so here.
But that line, I mean.
Yeah, it was like 22 maybe. Yeah, it was something. But also, for example, we have data from the previous year, and there was not the water was still colder, and we don't see any pattern. They just stayed in the depth. So here also is the good lack of observing mini heat wave in the data, because maybe now we tackle this fish and maybe we don't have any heat wave in 5 years. I guess we will have a bit of tricky. How many fish were there? Who gets closer will get the prize.
43.
No,
Almost 50.
25.
Higher.
78.
200.
So you were not paying attention to that.
To what you were saying.
So that's why I would measure that. You were listening to me, not seeing the presentation. Anyone, another guess?
84.
Sarah got it. 98.